In this lesson, we are going to look at gram staining. This method was developed by Christian Gram. Bacteria are differentiated by their gram reaction due to differences in their cell wall structure. Gram-positive bacteria stain purple with crystal violet after decolorizing with acetone alcohol compound. Gram-negative bacteria, on the other hand, stains pink with counterstain, that is safranin, after losing the primary stain, that is crystal violet, when treated with acetone alcohol compound. Quickly, let's go through the requirements of gram staining. That is grams iodine, acetone alcohol, and safranin. Those are the major requirements. Procedures followed in gram staining. Procedure number 1. Prepare a smear from the culture or from a given specimen. Procedure number 2. Allow the smear to air dry completely. Procedure number 3. Rapidly pass the smear 3 times through the flame. That is the uppermost part of the slide. Cover the smear with crystal violet for 1 minute and wash off with distilled water. Next, tilt off the water, then cover the smear with the grams iodine for 1 minute. Wash off with distilled water. Next procedure, decolorize rapidly with acetone alcohol compound for 30 seconds. Wash thoroughly with distilled water. Next procedure, cover the smear with safranin for 1 minute. Wash off safranin. Wipe the back of the slide and let the slide to air dry. Examine the smear with oil immersion objective to look for bacteria. Interpretation of the results. Gram-positive bacteria stains purple. Gram-negative bacteria stains pink. We go through our notes. Gram staining method was developed by Christian Gram. Bacteria are differentiated by their gram reaction due to differences in their cell wall structure. Gram-positive bacteria stain purple with crystal violet after decolorizing with acetone alcohol. Gram-negative bacteria stain pink with counterstain, that is safranin, after losing the primary stain or crystal violet when treated with acetone alcohol. Requirements are grams iodine, acetone alcohol, and safranin. Procedure. Number 1 is prepare the smear from the culture or from the specimen. Number 2, allow the smear to air dry completely. 3, rapidly pass the slide, that is the smear uppermost, 3 times through the flame. 6, smear with crystal violet for 1 minute and wash with distilled water. Tip off the water and cover the smear with grams iodine for 1 minute. Wash off the iodine with clean water. Decolorize rapidly with acetone alcohol for 30 seconds. Wash off the acetone alcohol with clean water. Cover the smear with safranin for 1 minute. Wash off the stain and wipe the back of the slide. Let the smear to air dry. Examine the smear with oil immersion objective to look for bacteria. Examine the smear with oil immersion objective to look for bacteria. Interpretation. Gram-positive bacterium stains purple. Gram-negative bacteria stains pink.